Friends, the artificial enzymes are the synthetic organic molecules prepared to recreate or mimic the active site of enzymes. Such artificial enzymes are often called as enzymes. The starting material for enzyme need not necessarily be a protein. It may be a material other than protein, like cyclodextrins, cyclophanes, palladium, and other star molecules. The binding of a substrate close to the functional groups in the enzyme causes catalysis by so-called proximity effects. So it is therefore possible to create similar catalysts from small molecules which will mimic the enzyme active sites. Living cells often synthesize complex molecules via multi-step sequential reactions, each catalyzed by an enzyme to allow all of the reactions to work well. Nature uses compartmentalization or site isolation, through which the individual steps are specially separated to optimize the action. The key to the achieving site isolation in solution lay in the use of star polymers, macromolecules that are capable of being functionalized so that they can bind and encapsulate small catalytic molecule in their core adjacent to the binding site for the reactant. A number of artificial enzymes have been reported catalyzing various reactions with rate increases up to the 10 raised to power 3. This is nevertheless substantially lower than natural enzymes that typically causes rate increase of about 10 raised to power 8. So what are the ideal requirements for the artificial enzyme environment? While constructing a new and complex nano environments which completely and closely surrounding the enzyme molecules should imply a strong chemical modification of the enzyme and possibly a very significant loss of catalytic activity. So keeping view in mind, a careful design of immobilization and post-immobilization techniques could provide a suitable procedure to create complex nano environments associated to minimal enzyme modifications. Covalent immobilization of one enzyme on solid supports having large surfaces with a given properties should provide a nano environment surrounding the area of the enzyme directly in contact with the support. Further, covalent immobilization or macromolecular polymer on the same large internal surface of the solid support should provide an additional nano environment surrounding the area of the immobilized enzyme molecules next to the support. Chemical modification of immobilized enzymes with polyfunctional macromolecules could also be an interesting way to greatly modify the enzyme nanoenvironment with a minimal chemical modification of the enzymes. These new environments surrounding the enzyme molecules should be mainly constituted by a fairly open structure that permits a substrate and products to diffuse towards the enzyme active centers. Agar-edges are composed by a 3D network of hydrophilic thick fibers 20 nanometer in diameter and much larger than the protein sizes, around 2 to 3 nanometer in diameter. Thus, proteins and other macromolecules become immobilized on large internal agar surfaces. Such supports geometry becomes a key feature in order to gel very intense enzymes support multi-point covalent immobilization. On the other hand, such supports geometry may be suitable to permit the adjacent immobilization of the polymers surrounding the previously immobilized enzyme. In this way, the co-immobilized hydrophilic polymer may fully protect a great portion of the surface of immobilized enzyme. Dextrans are very hydrophilic commercial polymers that are available with well-defined and very different molecular weights. In fact, dextrans contribute the polar phase of many aqueous two-phase systems, partitioning the organic solvent toward the other phase. Dextrans or their derivatives have been used to modify proteins, and they may be useful for chemical hydrophilation of both support and the enzyme surfaces. So the various approaches to design the artificial enzymes were developed. The traditional approach to enzyme mimic has focused on the de novo design of macromolecular receptors with appropriately placed functional groups. These catalytic groups are usually chosen to mimic the amino acid residues known to be involved in the enzyme catalyzed reactions. The realization of ideas in such a process can be arduous and although there are some impressive success, efficient catalysis rivaling enzyme rate accelerations still seems a long way down the line. Cyclodextrins may act as a Enzyme mimics. Cyclotonic strings, which consist of 6 to 10 glucose units linked head to tail in a ring. Glycosidic oxygen atoms and CH linkages point inwards, creating a hydrophobic environment which can act as a binding pocket. Catalytic activity is provided by the attachment of imidazole, hydroxyl and carboxyl groups as found in the active sites of serine proteases. The resulting enzymes resembles chymotrypsin in its esterase activity and furthermore is the more stable. Cyclorextrins may also be linked to a pyridoxal coenzyme producing enzymes with a transaminase activity. One of the most prodigious aspects of enzyme mechanism is the functional group cooperation often displayed in the active site. The electrostatic environment in the binding site maintains the delicate balance of pKs required for the various groups to participate in a catalytic fashion. In particular, histidine is often able to function as both an acid and base in a simultaneous bi or multifunctional catalysis. Basso and co-workers choose to mimic the enzyme ribonuclease. This enzyme uses cysteine and histidine as the principal catalytic group in the hydrolysis of RNA. To mimic this enzyme, two imidazole rings were attached to the primary phase of a cyclorextrin as the mimic-7 as shown in this figure. The mimic-7 catalyzes the hydrolysis of the cyclic phosphate 8 and shows the greater than 99:1 selectivity for the 8. This is a comparison to the simple situation reaction with NaOH, which gives a 1:1 mixture of both products. Isotope effects showed that the two catalytic groups were operating simultaneously and the pH rate profile, which was almost identical to the enzyme itself, shows that one imidazole functions in its protonated form while the other is unprotonated. The relative positions of the imidazole groups on the ring of the beta-cyclodextrin was found to be crucial. Only when the imidazole groups were attached to the adjacent sugars was a single product detected. This regular isomer of beta-cyclodextrin was not only more selective but also provided the fastest rate of hydrolysis and displayed the strongest binding to the substrate 8. Importantly, these results gave information about the mechanism involved, since the imidazole ion in this isomer would be better placed to protonate the phosphate anionic oxygen, which it can access more easily than the other catalyst isomers. The second material which is used as a artificial enzyme is the cyclopene. Another impressive applications of the design approach is a di-Aldrich pyruvate oxidase mimic. Pyruvate oxidase employs two cofactors, thymidine diphosphate and flavine, to water or alcohols to carboxylic acids or esters respectively by simple thiazolium ions in the presence of flavine. Di-Aldrich pyruvate oxidase mimic combines a well-defined binding sites with both the flavine and thiazolium groups attached in a covalent fashion, as shown in this figure. Proximity of the groups to the binding site and the intramolecularity of the oxidation step was therefore expected to improve catalysis relative to previous two-component system. It should also mimic the situation in the enzyme where the cofactors are bound in the enzyme active site, thus increasing the effective molarity of the reagents. Reversibly self-assembled dimers also mimic the enzyme activity. In a different approach, Rebeck have investigated the influence of a designed binding cavity on the rate of the disulfide reaction. In this case, no catalytic groups are required. Furthermore, although a disulfide is known, no natural enzyme catalyst has been isolated or is available for synthetic applications as yet. This is important since the desirability of designing artificial enzymes for reactions which have no convenient natural enzymes equivalent is self-evident. Rebeck and co-workers have carried out much research into the synthesis of reversibly self-assembled dimers. The dynamic behavior coupled with the microenvironment provided by the soft wall led Rebecca et al. Palladium can also act as a artificial enzyme. The palladium catalyzed reactions can be operated using these artificial enzymes. Hemine-based organic phase artificial materials also acts as a artificial enzymes. Based on the newly discovered artificial enzyme formed by mixing hemine with supramolecular hydrogels via cell assembly of amphiphilic oligopeptides, a novel organic phase artificial enzyme electrode by coating the artificial enzyme on an electrode which was then covered with a sodium alginate for protection. Scanning electron micrograph showed that the supramolecular hydrogel kept its nanomer structures on the electrode surface. Hemine dispersed in the supramolecular hydrogel as a monomer greatly promoted its direct electrochemistry behavior in organic solvents. At the same time, this electrode exhibited higher electrocatalytic ability to treat butyl hydroperoxide than pre-hemine model electrode. The biosensor can reach 95% of the steady state current in about 10 plus minus 2 seconds. More importantly, it can be applied in both hydrophilic and hydrophobic solvents without adding extra buffer or mediators to them that cannot be received by most traditional organic phase enzyme electrodes. This unique property greatly promotes the development of the organic phase enzyme electrodes by facilitating the detection of different kinds of substrates of the hemine-based artificial enzyme soluble in hydrophilic and hydrophobic solvents. Vitamin B12 can also act as artificial enzymes. A novel amphiphile having a arginine residue and a dihexadecyl moiety for the double chain segment was synthesized and its aggregate behavior was examined by electron microscopy, differential scanning, colorimetry, and fluorescence polarization measurements. The hybrid vesicles formed with alanine lipid and arginine lipid are morphologically very stable. Hydrophobic vitamin B12 derivatives which have carboxylic ester groups in place of the peripheral amide moieties of the naturally occurring vitamin B12 were incorporated into the single vesicles to construct artificial enzyme with vitamin B12 activity. In these hybrid bilayer vesicles, the enzyme mimicking reactions of methylmalonyl CoA muteins, which is 1,2 migration of carboxylic esters, group 1 axial ligand of the cobalt has been enhanced by microenvironmental effects. The antibodies also acts as an enzyme and accelerates the reaction by about 10 raised to power 3, 10 raised to power 4 fold. The field of artificial enzymes is a rapidly evolving subject as barriers between chemistry and biology becomes less distinct. A range of new methods which combine expertise from both areas are developing in recognition of both the fact that the de novo design approach can be time consuming and that a teeny miscalculation will be catastrophic. A trend in all these recent techniques is the use of selection approaches. The natural processes of selection and amplification is after all the way in which the enzymes have evolved their sophisticated function. Thank you.